Good morning, my friends. Molecular biology session today. Our eminent resource person, Associate Professor Dr. THIRUNAHARI UGANDHAR sir, working in the most reputed institution in northern Telangana, that is Kakatiya Government College Autonomous, Hanamakonda. Sir is going to explain one of the most important and also in all competitive exams related to molecular biology, biotechnology, biochemistry, environmental science, including botany, zoology, plant sciences, life sciences, CPGET, CU set, CPGET, CUET, and the AP set, TG set, CSIR net, and ARS net. Like all competitive exams, definitely minimum 2 to 3 questions we are observed in the area of the cell division. So Sir is going to start today's important topic, that is the cell division, cell cycle. So sir, please welcome and start your session, sir. Please welcome, sir.
Ready, sir. Okay. So today we are discussing the introduction of the cell division. So in the last class, which completed the chromosome structure, functions, types. So present cell division. This cell division is important for the growth and development of the living organism of plants and animals. In the plants, the cell division which can takes place in the two types of the divisions, mitosis and meiosis. In the mitosis, which occurs in the somatic cells of every living organism of plants and animals. The reproductive organisms, before fertilization, the pollen grains and ovules are formation from the microsporangium and megasporangium. So in the microsporangium, the microspore mother cell, megaspore mother cell undergoes meiotic division to form a reduce the number of chromosomes to form of the applied gametes, male and female applied gametes. The life formation starting meiotic division, continuation of the life division is mitotic. So both are which are responsible to the growth and development of a living organism and formation of the life, both. So this is also the first type of the cell division was reporting that scientists were cell. Cell is the four major scientists. The cell is the structure and functional unit of all living organisms. Are reported by Robert Hooke, 1635 and 1703. Then Anton von Leeuwenhoek was a microscopic organisms are discovered in the impossible without the discovery of microscopes. Then microscopes reconstruction and also observe under the microscopes, which are a small molecules, that is called animalculae, or bacteria, bacteria, fungus, algae, which are all is also microscopic, which can observe under the microscope. That is the unit of a all living organism. It is a microscopic and capable of independent existence. As the cell is a microscopic, the discovery of the cell is impossible without the discovery of microscopes. After microscope reported construction, it can starting off cell biology or cytology. So in 1665, Robert Hooke discovered cells while examining a piece of cork under magnifying lenses, maybe as a simple microscopes. After reported, Robert Hooke was observed under the cork, which under the microscopes, the cork, which consists of several number of compartments. Each compartment is also cell. He found that cork was made of a small chambers surrounded by walls. He called these chambers as cells. Was a first. So then second one, so these two scientists, and Schwann and Sladen, both are proposed the cell theories. the development, the structure, functional development of the all living organisms. That theory was are reported by the Sliden and Schwann, zoologist and botanist. So this is also pioneer of the cell biology. 1665, Robert Hooke coined the term cell after observing the Cork microscope. Then 1670s, Anton von Leeuwenhoek first observed the living cells. Examples are sperm cells, protozoa, and bacteria, which are all is also microscopic organism. In 1838, Matthew Sliden proposed that all plants are made up of the cells. Every cell which performing all metabolic activities, which are called the examples of the photosynthesis, respiration, nitrogen metabolism, growth, development, which are all is also metabolites, which can formation of the single cell of plant. Then 1839, Theodore Schwann proposed that all animals are made up of the cell, helping from cell theory. So in the 1855, Rudolf Wilshaw introduced a omnis cells or cellulae. All cells comes from the pre-existing cells. It is an important role of the genetics. The every cell which originated from pre-existing. The pre-existing qualities are transferred from parental to the daughter generation. That theory is also germ theory. Rudolf Virchow. Then Walter Fleming, first he described the mitosis and chromosome behavior of the cell divisions. Then also Edward von Bendham observed a reduction of the division to gametes. Early meiosis work is also reported the Bendham. Then Fleming is also called a mitosis. Both are called important divisions of cell. That is called as cell divisions. Then Wiesmann explained the meiosis emphasis is a role of inertancy and variations. The inertancy means it is the parental character features. Then also called various Koreans and race, etc. So the rediscovered the Mendel's work linking genetics and meiosis. It is also 19th century. The 20th century, various molecular biologists discovered the cell cycle. So this is also cell cycle means one cell divides to how much period, how many divisions, which can made to the single cell to two cells. That is called occurs cell division or cell cycle. So discovered the cell cycle control cycling or CDKs and also cancer mechanisms. So this is also cell cycles they reported in 20th century of our biologist. Then introduction to the cell division. Cells are the basic unit of life. By now, you know its structure and various theories are related to the origin of the cells. So that how to originate it from the new cells. These new cells are every period, every time, which can develop from the pre-existing cells. It have also been explained to you. In this unit, you will be studying one of the important process of the cell division. So the process of cell division may be different from unicellular and multicellular organism. In some simple unicellular organisms, one cell divisions may be enough reparative new organisms. Example, in say Pandorina, Eudorina, Euglena, amoeba, which are only single cell organism or unicellular organisms. It contrasts in other multicellular organisms. A large number of cell divisions are required to produce an organism with cellular complexity in the form of a various tissues and organs. Various tissues means structurally, functionally, developmentally, a group of the cells is called tissues. The tissues and tissue systems which can complexity form means that complexity means which are all functions. Examples of parenchyma, collenchyma, sclerenchyma, which are all is a group of cells, which are a particular character features are containing cells are packaged to formation of the complex tissues. These complex tissues are the examples of a xylem and phloem. compound tissues or complex tissues which are performing a different functions. One of the xylem is transfer of the mineral water from root to aerial parts of the plants. But other phloem is also transport of the organic solutes and solvents from leaf to all other parts of the plant. That is also two is also complex tissues or compound tissues. Then cell division is a process which one cell called the parental cell divides from the two new cells referred to as daughter cells. The first is also parental cell. So when the parental cells which can require to the energies or require to the nutrients, it can enlarge in size and divides too. That is cell division. And the cell division is identically similar to parental cell. That is mitosis. That each cell which performing structurally, functionally, genetically similar to the parent. Then the cell cycle is a repeating series of events that include growth. What is a growth? The growth is also increasing size of the organs and organisms like a leaf, shoot, and also called a fruit, flowers, which are cell divisions are major important for growth of the flower, growth of the fruit, growth of the seed. In even cell division is important for requirement to growth of the organisms. The cell cycle was discovered by Prevost and Dumas 1824 while studying these cleavage of zygote of a frog. The frog male and female gamete fused to form a zygote. That zygote frog cell division was reported by the Prevost and Dumas. It is the series of stages a cell passes through to divide and produce a new cells. Thus, the entire process with the help of a single parental cell in which a new cell population, growth, and development is known as the cell cycle. What is a cell cycle means? The cell population grow and development to the new cells, new organisms. That total part is called a cell cycle. So this is in plants and animals, meiosis is essential for sexual reproduction. The meiosis is important for the first formation of the life on the earth. That and the balance between applied and diploid stages are various. So during the applied stages, applied cells fusion, formation of the applied cell is called meiosis. Then development of the diploid cell is called a mitosis. The mitosis and meiosis are both are fundamental cell divisions for living organisms. So formation means that is a origin of life on the earth. This origin is comes from a fusion parts of two applied cells. That is male gamete and female gamete. So and the balance between applied and diploid stages are various. Fungi, aplantic life cycle, meiosis occurs just after gamete fusion. That is after gamete fusion, before gamete fusion. That is also two type. Aplantic life cycle, diplantic life cycle. Diploaplantic life cycle. That is so masses and ferns show alternation of generation. The sporophyte undergoes meiosis. The sporophyte phase, gametophyte phase, both are alternate to each other. In the sporophyte phase, diploid number of nucleus. In gametophyte phase, haploid number of nucleus. That is important. Higher plants and animals predominantly diploid. Meiosis occurs in gametes. Predominantly means the meiosis only formation of before fertilization. That is called a gametes formation. In the chlamydomonas shows zygotic meiosis, produce a applied juice spores from the zygote. So this is also applied. The applied spores, applied stages are more and diploid stages are less. That is called haplodiplanting life cycle, also called a gametophytic stage of plants. That is called examples of chlamydomonas, valvax, eugloina. That is all is also applied stage is a more stage is less. In human beings, diploid is a more stage than haploid is a less. Means at the time of the fertilization, before the gametes formation, in the total body is also diploid, but formation of the gametes is called haploid. That is difference of the aplantic life cycle and diplantic life cycles. The aplantic life cycles are examples of algae, fungi. Then diplantic life cycle is also examples of pteridophytes. Then bryophytes are aplodiplantic life cycle. So the cell cycle in eukaryotic involves two main types of cell division, mitosis and meiosis. Every cell. Then eukaryotes. The mitosis is an equitational divisions. Equitational means two equal, structurally, functionally, developmentally, two equal cells formation in cell division of mitosis, where daughter cells are genetically identical to the parent. Identical, same. It helps in growth, repair, and replacing old or dead cells. So old cells and dead cells are also called a differentiation. Old cells means parental cells. The new cells is also called daughter cells. Meiosis is reduction division that occurs in a reproductive cells. Like in a male, microsporange, female, macrosporange. Also flower, in the flower and ocean, gynoecium. But here in the animals, so that is called a ovary, and also called a ovary means that is called male gamete. Sorry, ovary is also female gamete which ovas are formation. Then male gamete testis. These testes are sperms formation. That is called male and female gametes are formation in the two different gamete, like testes and ovaries in human beings and animals. Other type of the plants, microsporange, megasporange. This microsporangium is located in the endoecium or anthers. Then gynoecium means ovary style stigma. In the ovary, the gametes formation is called a female gametes, macrogametes. These macrogametes which fuse to the microgamete to form a zygote. The cells produce a four genetically different haploid gametes with half the chromosome number during the meiosis divisions. But here mitosis structurally, developmentally similar to equal cells, parentally equal cells formation. So the was a Walter Fleming. The discovery of cell division is credited to the Walter Fleming, a German biologist who laid the foundation of cytogenetics. He was the first to systematically observe and describe the behavior of chromosome during the mitosis, contains the term mitosis. The first observation of the cell division during the division of chromosomes in the karyokinesis, cytokinesis, two stages. Then his work highlighted now chromosomes condense, elide, and separate during the cell division. First, then the chromosomes are thick, not elongated, not linear model. Before reporting of the Fleming scientist, after reporting Fleming, the chromosomes are condensed to at the time of the cell division, that is elongated tubular structures. That is called a chromosome formation. This was reported by Fleming. While earlier scientists had seen cell divides, Fleming's detailed and structured study marked a major advancement in understanding heredity and cell reproduction. Cell reproduction means one cell, two cells formation. That is called a mitosis. But understanding heredity means the genetical information, the genetical qualities or parental characters which can transfer from parental to daughter generation. That is called heredity. The heredity and reproduction both are important role to conducting of cell division. that is called a heredity and cell reproduction. But the importance of cell division can appreciated by realizing the following fact. The four important facts which is a important of cell division. The cell division is a prerequisite for continuity of life and forms the basis of evolutionary various life form. The evolutionary base, the cell is formation from uni. After unicellular organisms are evolved or originated to multicellular. That the cell division is a prerequisite for continuation of life. In unicellular organism, cell division is the means of asexual reproduction. The lowermost or primitive organism, only a sexual reproduction is there, not sexual. Example, Cyanophyceae class. In the bacteria, few species of bacteria are archaebacteria. The Cyanophyceae, the total sexual reproduction is absent, means it is a most primitive character of organism. In unicellular organism, cell division is means of a sexual reproduction, which produce a two or more new individual from the mother cell. The group of such identical individual is known as a clone. The clones are formation. In multicellular organism, life starts from a single cell called zygote. The zygote formation from two applied gametes of male and female. Then after egg fertilized to zygote, it transfer into the adult that is composed of the millions of cells formed by successive divisions. After meiosis division, the zygote undergoes mitotically, not meiosis. Meiosis are only takes place in a one time of life formation. Then life continuation is called a mitosis. This is also two different things. Cell division is the basis of repair and regeneration of old and worn out tissues. Old tissue and worn tissues both are which are divides. So this is a important. Then the cell division is cell reproduction or cell multiplication is the process of formation of new or daughter cells from or pre-existing of the cells. It occurs three ways of the cell divisions. Mitosis means are direct cell divisions. Mitosis are indirect cell divisions. Meiosis are reduction cell divisions. It is also the three type of the cell division is proposed to the genetical or hereditary scientist. These hereditary scientists, examples of Mendel, also Fleming scientist, was proposed the mitosis, mitosis, and meiosis. These are three cell divisions. But amitosis, this amitosis means direct cell division. Greek word a means known. Mitosis means thread. Asis means thread. State. Mitosis is mode of division in which nucleus elongates, constricts in the middle and divides directly into the two daughter cells. Mitosis means not performing all stages of mitosis. Only one stage which can divide two cells. That is mitosis. This is followed by a centripetal construction of cytoplasm to form the two daughter cells. It is not similar, not structurally equal. Unequal cells are formation in the mitosis. Mitosis means structurally similar to each of the cell. But here is also diagram represent of the mitosis. That mitosis, which is also that single cell of the bacteria that is a divides in the nucleus and it can formation of the two cells formation. These two cells are which is also structurally, functionally similar to the parents. That is, a mitosis occurs This mitosis is important type of the cell division which can occur in the bacteria. But mitosis characteristic, it intact nuclear envelope found through not the division. First point, the chromatin does not condense into the definite chromosomes. Only chromatin is there. That chromatin is not directly divides into the new chromatids to passes to the new cells or daughter cells. The third one, a spindle is not formed. That is important. Mitosis and mitosis. Then the fourth one, the chromatin distribution occurs unequally, which causes a abnormalities in metabolism and reproduction. The metabolism and reproduction is also abnormality, also differentiations. Then cytokinesis may or may not follow karyokinesis. After karyokinesis, cytokinesis. This is also important for the mitosis. But here, mitosis, the cytokinesis and karyokinesis are not one above the other or alternate to each other. That is differentiation. The cytokinesis may or may not follow karyokinesis. Mitosis occurs in a meganucleus of paramecium nuclei of internodal cells of charm, endosperm cells of seeds, endosperm seeds, then cartilage cells and diseased cells. Remark 1955. Remake scientists discovered the mitosis in RBC, red blood corpuscle. First reported the remake scientist in 1955 mitosis of chick embryo, but the term was coined by the Fleming. The mitosis was coined by the Fleming. Mitosis was remake 1955. So this is a mitosis, but here is also mitosis. But this is also called the stages of the cell divisions of the mitosis. The mitosis which occurs here, G1, G2, S1. G1, this is also first one, G1 means growth 1. Growth 1, RNA and protein synthesis, cell growth, nucleolus. Then after G1, then also called cytokinesis are there. Then G1 also called S phase, DNA synthesis, histone synthesis, and central S after G2 phase. G2 phase means multiple of cell organs, cells, growth, nuclear growth, biochemical for M phase, which is called a prophase, metaphase, anaphase, telophase. These are all is also karyokinesis. Then after cytokinesis to formation. This is also called M phase. M phase means here is also G1 phase. These are S stage. This one G2 stage. This one M phase. The 4 stages, which is called a cell cycle, means one cell divides the 2 cells. The mitosis are indirect cell divisions, equational cell divisions. Mitos means thread, asis means state. The synonyms: indirect cell division, somatic or